Mike, thank you for taking time to be part of our gathering here today. It's good to have you with us, and good to have that information as well. Speaking of which, we are now going to be joined by Dr. Emmeline Hill of Plusvital, and very curious to hear this presentation as we move into the area of genetic testing and related subjects. So, Dr. Hill, if you would, please.
Good afternoon. Thank you very much for the invitation to be here today to talk to you about a relatively new area of interest to the thoroughbred industry, which is the area of genetics. I am here representing Plusvital, which is an Irish equine science company that was founded in 1975 and which merged with Genome, a genetic testing company that I established in 2010 with the Irish trainer and breeder Jim Bulger. That company developed as a university spin-out company from University College Dublin, where I'm a researcher, after we made a discovery which is now referred to as the speed gene. We made the discovery of the speed gene quite soon after the horse genome was sequenced in 2007. The sequencing of the horse genome really was a revolution in equine science, which has really changed the science of genetics for the horse and is changing breeding in a way that it probably hasn't seen for hundreds of years. I'm joined here by our representative in America, Stephen Mulvaney. Stephen's based here in Kentucky. And if anybody's interested in commenting or has any questions, please use our Twitter at Plusvital and include the hashtag SpeedGene, and we're happy to respond to your questions that way, or we can answer them at the end of my presentation. So we currently are in a period of great change for the thoroughbred industry. In other livestock production industries, such as cattle and pig breeding industries, the systematic application of genetics has completely transformed these industries. For example, milk yields have quadrupled in dairy cows in the last 15 years. And this is by applying the science of genetics to assist decision-making processes in breeding, using what is known as marker-assisted selection, using information from the genome to help select individuals for the next generation of breeding. These new technologies are indeed challenging the way that we think and how we're making decisions now about our own bloodstock. So how has the genome sequencing changed really what we're doing? When we refer to the horse genome sequence, what we're referring to is the 3 billion letters of the genetic code that make up the horse. I'm going to explain a little bit about that later. Almost all of these 3 billion letters, which is the information that makes a horse, are shared among thoroughbreds. So almost all horses have exactly the same 3 billion letters, which makes them a horse. But there are subtle differences, and there are a number of differences in the genetic code that make each individual an individual. It's why you look different from your brother or sister, and why one horse, a full sibling, may be different from its full sibling. So it's not actually the understanding or unraveling the sequence of the genome that was revolutionary, but really understanding the differences in the genetic code and how that applies to and manifests itself as different traits in the horse really is what is allowing for more strategic and informed decision-making today. These new technologies are not without controversy. The International Thoroughbred Breeders' Federation have explicitly stated that they are not interested in mainstream genetic testing, which is coordinated and a coordinated and cooperative effort, as has been seen in other production industries. There's some concern that perhaps a mainstream approach like this might change the pattern of racing and change some values. However, they have said that individuals may use this or these sorts of tools in individual operations and may be to their advantage and may be beneficial in areas such as health. Also, among breeders, some things that we hear from people that we encounter in the industry are things like, well, this might ruin the industry in some way, which I'm not entirely clear how, but I'm going to address this later in my presentation. In fact, all of these points, which I think may be of some concern. Some people have said, I've been successful up to this point. Why would I change? And in fact, I think that's one of the difficulties with change. I think people have a recipe and a successful recipe, a successful way of doing things, and you start to introduce something new. How is that going to affect the status quo? Will it affect the status quo? There's a lot of scientific research that has been published in this area from my group and other groups around the world. But the question often comes back to us is, okay, well, that's fine in the scientific realm, but does this actually really work in practice? As opposed to in principle. And I'm going to give you some examples of how it does, how it may be applied in practice and how it does work. And from your perspective as breeders and consigners, the question we often get is this going to actually make it harder for me to sell my horses? And how am I going to benefit? So what's all the fuss about? Well, genetics is the field of science which is involved or in understanding heredity or components that are passed down from one generation to the next. When we refer to the genome, we're referring to the complete complement of genetic material that's contained within the complete set of chromosomes. And horses have 32 pairs of chromosomes, one of each pair inherited from the sire and one from the dam. So when we refer to the genome, we're talking about the complete complement of DNA that's passed down from one generation to the next. But of course, whether it's recognized or not, people have been using genetics for 300 years breeding thoroughbreds. Because anytime you're looking at a pedigree, you're trying to evaluate the genes that have been passed down from ancestors in that pedigree to that individual that's represented in the pedigree. And really, that is trying to understand genetics. It's trying to understand the genetic material that's been passed down from those ancestors. It's important to point out when we're thinking about genetics is that the value of genetics is proportional to the amount of variation in a trait that can be attributed to or accounted for by genetics. This is something that we refer to as heritability. If the heritability of the trait is high, then the value of genetics towards understanding that trait is going to be high as well. The heritability or the genetic contribution to performance, depending on how you define performance, and of course this is something that we would all struggle with, whether we're defining performance as being a group performer, whether it's earnings, whether it's the time of the race or handicap rating. Scientific studies have estimated that the heritability or the genetic contribution to performance traits ranges somewhere from 30% to 50%. So that's quite a considerable amount, but it's not the entire complement. The rest of, I suppose, the other contributions to performance is the environment that individual encounters in its lifetime, from, I suppose, the in utero environment to early stages in its life, all the way through managing the young horse, and then managing the horse when it's in the hands of the trainer. All of these influences affect the individual and affect the individual's chances of success. And because genetics is only a fraction of that total success, in no way will genetics ever replace the skills of the horseman and the horsemanship that is required to produce an elite athlete. What it is that it's a part of the puzzle, and it's the part of the puzzle that we're trying to evaluate when we're looking at a pedigree. Now, some traits have a higher heritability than others, and this is where I mentioned that the value of genetics is dependent on the heritable component, and it's something to bear in mind as I go forward. Some of the challenges that breeders face is that they want to produce horses at sales that are precocious looking and well presented at the time of sale. And generally this would be at yearling sales. And sometimes this is a challenge. So while you may have the horsemanship skills, the genetics may not always contribute to the enabling of a horse being ready and presentable for sale at the stage that you're interested in, or that you're trying to sell your horse at. Other challenges for breeders is having high return rates, and also a major challenge is to manage mating decisions. When you're having to make a decision on stallions and trying to predict what the next fashionable stallion may be, and particularly trying to select stallions that may be unproven stallions or young sires, and trying to best guess at that stage what sort of horse is that stallion going to produce, and is that stallion going to be compatible with your mare. So with all of the tools that you have in your toolkit to date, you can use all of those to try to produce horses for sale. But we can also help by applying genetics into the mix, by enabling scientifically sound or introducing scientifically sound data to more strategically inform the decisions that you're making at each stage. In particular, you can use genetics to identify your best breeding prospects from your broodmare band. Plan your matings to more consistently produce the types of horse that are going to be of higher value at the sale, and overall maximize your chances of success. And I'm going to tell you how you might be able to do that using some of the tools that we can provide as scientists now, and particularly in genetics. One of the most effective breeding tools, or genetic breeding tools, that has been developed to date is arguably the speed gene, or we now refer to as the speed gene, which has been validated in numerous scientific studies, and has been shown to be strongly associated with the optimum race distance of an individual horse, and is highly predictive of the type of horse, highly predictive of the type of horse that individual is likely to be. I'm going to explain these in more detail, but what we refer to as CC horses, we refer to CC horses, these tend to be sprinters, or tend to be more early maturing individuals that are suited to short distance racing. CT horses tend to be the middle distance type horses, and TT horses tend to be the longer distance types. And I'm going to explain how this works now. The speed gene is actually a term that we coined for a gene that is known as myostatin. Now in the horse there are 25,000 genes. Myostatin is just one of these 25,000 genes. When I started doing this research in 2005, 2006, we knew in a range of other species that mutations in the myostatin gene caused very unusual muscling characteristics in other species. The animal on the left is a Belgian blue bull, and he has what is known as the double muscling trait. He looks like that because he has 2 copies of a mutation in his myostatin gene, one that's inherited from his father and one that's inherited from his mother. And what happens is myostatin is a negative regulator of muscle mass development. When myostatin is mutated and it's not produced, then it can't control muscling, and muscling then goes out of control. And this is what we see here in this Belgian blue bull. The dog in the middle, believe it or not, is a whippet. It's what is known as a bully whippet. And he looks like that because he has 2 copies of a mutation in his myostatin gene, one inherited from his mother and one from his father. Now that's actually detrimental to racing performance for this dog because he has too much muscle bulk. But dogs that have 1 copy of the mutation inherited from either their mother or their father are better racing dogs than dogs that have no copy of the mutation at all. And these mice on the right, the bigger mouse on the left is known as a mighty mouse. He's a genetically engineered mouse that actually has no myostatin at all. And that's why he looks like that. So we knew all this from all of these other species. And when I was doing starting my research back in 2005, I thought, well, are there any differences in the thoroughbred? Are there any differences in horses in the myostatin gene? And that's why we started looking there. Nobody had published anything, and there wasn't anything in the public domain. So what we did is we set about reading the letters of the DNA code, or sequencing the DNA code of the myostatin gene in the thoroughbred. And when we did this, we found that there was a difference in the gene, in the myostatin gene, that changed a letter of the DNA code from a T to a C. There are 4 letters of the DNA code, G, A, T, and C, and it's combinations of those letters that make up the genome, and there are 3 billion of those letters. Those are the 3 billion letters that I referred to earlier. So what we found is that if you imagine this blue bar is a chromosome, and the orange block is the gene, the myostatin gene, there's a particular sequence of letters that spells out to the cell to make the myostatin protein in the cell. And at a particular position, there can be a letter T, but on other chromosomes, they might have, at the same position, a letter C. So this is essentially a mutation, or also known as a DNA variant, that is where there is a letter T on some chromosomes and a letter C on other chromosomes. What this means is that when an individual inherits a C or a T chromosome, that makes up their genetic type. So if an individual passes on a T-type chromosome if an individual inherits a T-type chromosome from the sire and a C-type chromosome from the dam, then their genetic type is what we refer to as CT. If an individual inherits a C-type chromosome from the sire and a C from the dam, then their genetic type is CC. And if they inherit a T from the sire and a T from the dam, then their genetic type is TT. I should say that they're also CT if the T comes from the dam and the C comes from the sire. So all thoroughbreds, in fact all horses, must be one of these 3 genetic types of the myostatin gene. They must be CC, CT, or TT. When we looked at the distribution of these 3 different types in the racing populations, we found that there was a strong association between the genetic type and the distance that a horse is best suited to racing over. And this was done initially on a couple of hundred stakes race winners, but we have since tested 15,000 horses, and 95% of the horses that we've looked at that we know their race records fall very clearly into these categories. These are the distances for horses in North America that we've tested, almost 1,000 of them to date, that are race winners, I should say. So CTs tend to perform better over the shorter sprint distances, up to a mile. The CTs are more versatile in that they can perform well as 2-year-olds and then train on over the longer distances as they have the opportunity. And particularly, this is very relevant in Europe, where this can occur. And then the TT horses are better suited to longer distance races. Some people, in fact, many people for years, until this was available, until the genetic test for distance was available, pedigrees were used to try to determine whether individuals were best suited to different types of racing. Not only the pedigree, but also the physical evaluation of the individual. What the horse looked like, how it performed under saddle, before a judgment was made in terms of the race type that horse would be suited to. And pedigree is a reasonably good indication, but it doesn't actually tell you everything. And I'll show you why. You may, if you cast your mind back to high school days, remember these Punnett squares, which you may have learned in biology class, referring to Mendel's peas, and trying to predict whether there'd be curly peas or round peas from a particular cross. But I always call this Mendel's peas in real life. So if you have a mare who's a CT mare, and you take that mare to a CT stallion, and imagine you take that mare to a CT stallion 4 years in a row, what are the types that you're likely to produce? Well, in fact, you're not going to produce CTs every time. What happens is that only one chromosome is passed down, so the mare can only pass either her C or her T on each time, and the stallion can only pass his C or T on each time. So what happens is this. If a C is inherited from the mare and from the sire, then a CC foal is produced. And if, you can see here the different combinations then being passed on. So what happens is that all of those 4 foals have identical pedigree pages, yet genetically they're very different. 25% of the time, a CC foal will be produced, 25% of the time, a TT foal will be produced, and half of the time, a CT foal will be produced. And this has a very stark influence on how that horse performs subsequently performs on the racetrack, but also we found has a very big influence on how that horse matures as a young individual and how it shows at sales. And I'm going to show you that data now in a second. So if knowing this, what we know then for individuals that are the CC, CT, or TT types, they can only produce certain types. So the CC horse, whether it's a mare or a stallion, will only ever produce CC and CT foals because that individual doesn't have a T to pass on. If your mare or your stallion is a CT, then it can produce a CC, CT, or TT foal depending on the stallion or mare, depending on the mate choice, and also depending on what's passed on. And a TT individual will only produce CT and TT types. So once you understand how this works, it's important to be able to identify the opportunities for each genetic type and to tailor your breeding programs accordingly in order to more consistently produce the type of horse that you desire. So the first thing is to identify the opportunities for each type of horse. In North America, obviously, there's a difference between dirt racing and turf racing. There are 70% of stakes races on turf are run over distances of less than 8.5 furlongs. That's compared to 11% of stakes races run over distances of less than a mile on the turf. So there are clear differences in opportunities for the CC, CT, and TT types on both of these surfaces. When we look at the distances and the distance opportunities on these surfaces, we find actually that on turf, each of the genetic types on average has a distance about a furlong greater than it would have if it was raced on dirt. And that's because of the different energy requirements, the different requirements for racing on dirt and turf. But you can still see within each of the dirt and the turf categories a very clear distinction in the different race distances for each of the different types. What we found, even though we weren't looking for this originally, we hadn't hypothesized it per se, was that these different types manifest very differently in terms of how they develop as young horses. The CC types are significantly more precocious in their development than TT types. And we can see this reflected then in their earnings in the sales ring and their earnings on the race course. So you can see here the average two-year-old earnings for CCs, CTs, and TTs, and their average lifetime earnings. So as two-year-olds, the CCs and the CTs by far outperform the TTs. But later on, the TTs have an opportunity to catch up as they develop and as they mature, and as the opportunities arise for them to compete over the longer distances. The reason for this is that we have shown in studies in collaboration with researchers in Japan and also here actually in the University of Minnesota, that the myostatin gene in the horse is having two effects. One, it's having an effect on the rate of muscle hypertrophy or muscle growth in the young horse. And the other thing is that it's having an effect on the laying down of the muscle fiber types. CC horses have 7% more muscle mass or greater muscle mass than TT horses in the middle of their two-year-old season. They also have almost 10% more fast-twitch glycolytic fibers than TT horses. TT horses tend to have more slow-twitch aerobic muscle fibers that are more suited to endurance type racing. So these are the physiological changes that occur, and these then manifest themselves on the race course, as I've shown you here. We collected samples with owner's consent from a sale 2 years ago, a horses in training sale, a 2-year-old horses in training sale, where the horses breezed or did a show under tack at the sale. What we showed, or what we found, is that the CC horses on average achieved a significantly higher sales price. And this may or may not be because they had a significantly faster breeze time than the CT horses and the TT horses. Now, 13% of the horses in the sale were TTs, but I think you can clearly see if you had a CC horse at this sale, you were going to achieve a higher price, and probably because of the way the horse looked and also how it performed in the show under tack. We also did a similar study where we looked at a set of yearlings that were sold at sale in Australia. These were horses that were collected in advance of the yearlings, of a set of yearling sales in Australia, the Magic Millions sales at Inglis Easter and Melbourne Premier sales. So what we found here is that the CC horses, again, had a higher clearance rate and achieved significantly higher sales price than CTs and TTs in the sale. The CCs received over almost $26,000 more on average than CTs, and $66,000 more on average than TTs. Why do CCs tend to outperform TTs at point of sale? Well, I think the explanation for this is that this myostatin gene is having a significant effect on how the horse is developing. So as it comes to the sale and it's more developed, it's looking like it's ready to perform and go out and perform as a 2-year-old. And at these particular sales, especially in Australia, I might add, where there's an emphasis on early 2-year-old performance and sprint performance, that's what people are looking for. And that's reflected then in how these horses are being returned at sale. So we would contend is that as a breeder, if this is the type of horse that you're trying to produce for sale, then you can increase your chances of success by more consistently producing this type of horse through active management programs and strategically selecting mates for your mares on the basis of this genetic information. So if you want to produce a precocious 2-year-old type, the type of cross that you should be looking at doing is, if you have a CC mare, avoiding the TT types and selecting stallions that are CC or CT types. And this is just an example of ensuring that you're going to produce CC and CT types, because as I showed you earlier on, a CC is never going to produce a TT type. It works the other way around, whether it's if it's a stallion or a mare being the CC or the CT type. I always say that breeding is like a genetic lottery. You never know what's going to be passed down, but what you can do using this information is essentially to load your dice. So if you have a CC mare and you want to ensure that you're producing CC foals,